lunch topic. So our next guest presenter, Andrea Taddei, will be telling us about identification of fruit fly larvae of non-European Tephritidae intercepted in the European Union. Please.
Hi everybody. I'm glad and grateful to be here these days in the place on earth with the highest concentration of enthusiastic Diptera workers. This is very inspiring for me. So thank you. My name is Andrea Taddei. From the Plant Health Laboratory of the French Agency for Food, Environmental and Occupational Health and Safety, based in Montpellier, southern France. In 2019, our unit in consortium in partnership with the Austrian Agency for Health and Food Safety in Vienna became a European reference laboratory, meaning that it coordinates the network of 27 national reference laboratories of the member states by supporting them in the and providing guidance for the identification of regulated arthropod plant pests. And among these regulated quarantine pests in Europe, there are, of course, fruit flies. Tephritidae are very frequently encountered at import control in Europe as larvae infesting fruit and vegetable consignments. In the period 2014-2018, Tephritidae were the most frequently intercepted harmful organisms in Europe. Until recently, according to previous regulation, identification of fruit flies at import control as non-European Tephritidae was sufficient to trigger phytosanitary measures, thus to reject the consignment. However, since 2021, this is no longer the case, since a new regulation amended the previous one and introduced a big changement. The general group of non-European Tephritidae was replaced by a list of 75 Tephritidae taxa, genera and species, after a big work of risk assessment by the European Food and Safety Authority. What are the consequences of this new list? That now national reference laboratories, plant health laboratories, are expected to be able to identify these pests at the genus or species level, which is very challenging, I would say at present impossible. Why? Because you know that better than me, identification of fruit fly larvae is in itself tricky, and it depends, of course, on availability of larva description. It depends on the larva stage available and on the experience of the staff. Usually, description is done by CAM imagery, and this equipment is usually not available in diagnostic laboratories. And also for this new list of taxa, there are, for the vast majority of them, there is no information about larva morphology and no barcodes are available. So let's have a quick look at this list of regulated fruit flies whose introduction is spread over the European territory must be prevented. Here is the list. I put in red the taxa which are well known to be of significant importance and for which morphological identification of larvae may be possible in some cases. For the rest, larva identification is really hard. I put here just 4 examples. But represented the vast majority of this list for which there is no description of larvae, no CO1 barcodes. Thank you, Valerie, for the picture of the wing of Urophora christophi. We couldn't find anything in the web, in the literature. So this of very valuable reference literature and web-based applications. A suitable, comprehensive identification tool for our purpose is missing, especially for larvae. So how can the European lab help with this? We conceived a dual strategy. First step, to design an online matrix key, including all the 75 taxa from the new regulation, for both adult and larvae, when possible. Second, try to unveil what we call the black hole of non-EU Tephritidae intercepted. In other words, to investigate intercepted fruit flies in Europe over time, and only identified and reported and non-EU Tephritidae, which was allowed by previous regulation, but we don't know what's hidden behind this. Why to do so? To build a clear picture of taxa that are actually intercepted in the EU compared to the new regulated list, and assess the possibility to develop a rapid, simple, and reliable morphological identification tool. So we started by analyzing data available in our laboratory in France. I have to say that actually in the last 2 decades in our laboratory, identification of fruit fly larvae had been quite accurate, thanks to the rearing to adults and more recently thanks to barcoding, CO1 barcoding. To go a bit further and try to see if any taxa had been unnoticed in the last 25 years. So we selected a number of fruit fly specimens to submit to a first morphological characterization, followed by a non-destructive DNA extraction and CO1 barcoding to confirm identification, to actually know what it is, and followed on its turn by a second morphological characterization after slide mounting. After that, as a second step, we wanted to involve our network of national reference laboratory, and we asked them to share fruit flies import data for their respective country and fruit fly specimens available in their working collection, so that we could perform the same morphological and molecular characterization as for the specimens from France. As a third step, we wanted to assess the possibility of identification for those taxa. So we selected a set of characters which are visible at the stereo microscope, at the compound microscope. And we also associated to this taxa the pathways, meaning the countries of origin of the commodity and the host commodity. All of this with the aim to get to simplify key within the larger Q-Tephritidae key under development for the species, only for a subset of species that are which are those that are actually intercepted in Europe. So we can say that a relatively limited number of taxa are intercepted, have been intercepted in Europe over the past 25 years, some of them repeatedly, some of them only occasionally. They are all regulated pests except for Ceratitis capitata, which is no longer regulated in Europe, Neoceratitis cyanescens, and Rhagoletis cerasi. And they mainly belong, which is not surprising at all, to the 5 big genera of economic importance: Anastropha, Bactrocera, Ceratitis, Dacus, and Zeugodacus. For those taxa, morphological identification, at least to the genus level, is possible when host plant and country of origin are known. The main characters that we selected are the presence or absence of accessory plates, presence or absence of dental sclerite, number of tubules on anterior spiracle, presence, absence, and size of preapical tooth, and distribution of dorsal spinulae. So let's have a look at those intercepted taxa based on the macro geographic area of export, let's say. I will not go very much into detail about the morphological characters because I don't have time, but we can discuss this later if some of you are interested. So from Central and South America, not surprising again, we encountered, we had Anastropha species and Ceratitis capitata. Within the Anastropha genus, we had ludens, striata, and the Fraterculus group, but we don't go further in the identification, and I'm very excited to learn here today that new tools, morphological and molecular tools are under development. Anastropha species can be separated from Ceratitis capitata by the presence of accessory plates, which are not present in Ceratitis capitata. Also, the dental scleroid is present in Ceratitis capitata, but not in Anastropha ludens and in Anastropha species. Sometimes also the concave shape of the anterior spiracle may be useful to identify Anastropha. From sub-Saharan Africa, we had several Ceratitis species other than capitata, Cosira, the FARQ complex, and Quinaria. They are all characterized by a small preapical tooth, which is not the case for Dacus species, which display a much larger preapical tooth. We had Bivitatus, Ciliatus, punctatifrons, and probably we will soon analyze some samples of a likely Dacus armatus. And two Bactroceras species which are invasive introduced in Africa, Dorsalis and Latrifrons, we can be easily identified. And Zygodacus cucurbitae on Cucurbitaceae. Also, Ceratitis capitata have been intercepted frequently from Africa, but it can be easily distinguished by all the other taxa due to the lack of accessory plates. From Asia, again, several Bactroceras species, Correcta, Dorsalis, Latifrons, Minax, and Zonata. Some of them cannot be distinguished between each other. A bunch of Zygodacus species, Caudatus, Cucurbitae, Tau, and Lineatus, and Zygodacus ciliatus as the only Dacus species that have been intercepted so far, based on our data, of course. So, to sum up, around 30 species are intercepted in Europe. It's not still, I say around because it's not still defined. We are still working on it. These taxa can be identified morphologically, at least at the genus, to the genus level, if of host plant and host origin, is known. Within the larger Q, Tephritidae key under development, a smaller key for actually intercepted taxa is under development. It's meant to be a dynamic tool, which will be constantly updated each time new data are available. For example, new taxa are intercepted, some characters that are updated due to higher variability observed, or new import pathways. So this is a simple morphological tool for rapid identification at import control, but CO1 barcoding still plays a key role in validating the morphological identification, and also rearing to adult tooth when possible is even better and highly recommended. Last thing, we detected quite a few inconsistencies with the literature about some characters, especially for the distribution of dorsal spinulae for some taxa and about the number of tubules on anterior spiracles. So I want to thank my collaborators and all of you for your kind attention, and I'm happy to hear your comments or try to answer your questions. Thanks a lot.
Thank you, Andrea. And yeah, we do have time for questions. Please, Alan.
I don't suppose you had anything to do with developing.
No. This is a... Actually, there is the... We were puzzled about this list. It's quite surprising. It was quite surprising for us. But there is a pest categorization by the European Food Safety Authority. So, what can I say? What can I say? Yeah, there is probably no risk or no possibility that Europhora christophi became invasive and poses economical problems in Europe. But what I say, from my point of view of diagnosticians, is a big problem, because we are expected to be able to identify these pests to be ready to promptly detect them, if in the remote case that they come to Europe. But we're trying to overcome this problem by, as I try to explain, to really focus on, really know what we actually intercept, to know what we actually intercept, so we can just focus on those and try to get around the problem of lack of identification methods for this taxa.
Please.
Actually, I have a comment for this pest categorization by EFSA. So the approach was to include any species that might have an impact in the European sector of plant, the major pests, that even for ornamental pests, for everything. So that was the approach. Mm-hmm. It was like categorization is 250. That this was the approach was to include everything and then from there to narrow down.
I'm not questioning the list, of course. I don't have any experience about pest categorization or risk analysis. I'm just saying it from my point of view. I'm trying to tackle this problem of lack of information about this taxa.
Alan?
Based on details. And I think that's why I always has a study.
Sorry, I didn't understand. I mentioned that.
I say we can probably provide some advice on the details of what you're talking about here, especially.
Yep.
For example, the mouthhook of medfly doesn't always have.
Yeah. But I think I haven't mentioned this character for medfly. I just mentioned the absence of accessory plates. And the presence of the dental sclerite in comparison with the Anastraphus species, just to differentiate with those. And medfly can have a small periapical tooth in some populations, but I have not mentioned that in my presentation. Yes.
Well, thank you again, and we'll continue after lunch. Thank you for attending.